It's my pleasure to introduce Dr. Archibald, Chief Scientist, VP of Scientific Affairs, President of the Integra Foundation. He joined Integra in 1997 and oversaw the development of Neurogen, a nerve guide system. And in that same year, started a program to develop DuraGen, a dural graft matrix for wound healing. His numerous accolades include assistant professor at Duke University, a fellowship at Harvard University, a PhD from North Staffordshire in the UK. Again, I welcome you, and the title of his presentation, as you can see, is Boats, Fish Skin, and Regenerating the Nervous System. Thank you.
Good afternoon, everyone. Thank you for coming to the talk. And thank you very much, Kathy, for the introduction. It's always embarrassing to hear yourself being introduced so nicely. So today I'm going to talk a little bit and introduce myself and my career. That accounts for a somewhat cryptic title of the talk. And then I'm going to really sort of concentrate on what we've been doing over the last 20 plus years in translational medicine, in regenerative medicine. And then please, at the end of the talk, there are no holds barred on questions, both from career, motivation, and what we're doing currently for research and development and product development. So early influences, and I think, not many, we sort of discount them sometimes in our careers. But I was very fortunate to grow up on the edge of the ocean on the east coast of England. That had a profound effect on my interest in natural history and the environment and anything which was alive, whether it was plant or animal. And that sort of really came to the fore, not just in learning to swim early and snorkeling and fishing and learning how to row a boat before I was 8. In starting to practice falconry. I used to rescue all sorts of injured birds and seagulls and stuff off the beach, and one day somebody brought me a hawk, and we managed to raise it and then figure out that we needed to train it, to teach it how to hunt, and trained it and then released it. And I was 11 and 12 years old at that point, and that really gave me a passion for birds of prey, and particularly, and learning a great deal about their biology, welfare, healthcare, and training. And I think it sort of set me up for a career in biology, even though I didn't pursue, which is always strange to me, but I didn't pursue that branch of biology as a career. However, the boat, scuba, and fish, I think is for more pragmatic and understanding how the world works. I think I learned a lot of physics from scuba diving, which I was then able to understand better in high school. We had a lot of physics in the UK education. Boats give me both practicality in sailing, I think, and have gone around the UK a couple of times and down into the Atlantic and up into the Baltic. In my late teens and early 20s, crewing for various people. And then just fishing. Fishing was a passion, the ocean was right there, but both that and the biology of fish, and also for oyster culture, really was very interesting to me. And I did my second year thesis at zoology on oyster culture. And so I went to do a zoology degree. And I came out with being fascinated by biomechanics and neuroscience both. And I did a PhD in basically in neuroscience, but in a biomechanics department. So I was sat between two departments, one at Keele University and one at North Staffordshire University, and studied the properties of peripheral nerves and how they react to surgery. And then tested different types of surgical manipulation to repair nerves and looked at the functional outcomes and the histology. And you'll see a few pictures from way back then in the talk later on. I was lucky enough to get a postdoc in the UK. The environment is very different today, but in the mid-80s, was a very reduced funding for biology and medicine in the UK, and I was very lucky to get a postdoc in postgraduate medicine at Keele University, and that turned into a fellowship at Harvard, which was, I think, a little unexpected to go from the Midlands in the UK to a post at Harvard. But that really changed my career. I left the UK and I intended only to be here for a couple of years, and it's now been nearly 35 years. So I guess I'm going to stay. But then we went from the fellowships, did another fellowship at Duke University, and I went on faculty at Duke for a while in the Department of Neurosurgery. And we followed cellular basis of peripheral nerve regeneration and axon guidance during regeneration, which applies both somewhat to the CNS as well as the peripheral nervous system. And we had worked on a collagen nerve guide. I was working on intubulation repair in my PhD and postdoc in the UK, and we continued that at Harvard within another team, and actually developed a device which could be implanted. And I took a sabbatical in '97 to take that into the clinic. And that was really, I came to the US thinking to be only here 2 years. I went to Integral Life Sciences thinking I'd only be there 2 years and back to academia. And that too has been 24 years now. So I think when people look at careers like this, it looks like a straight arrow. I think that's a myth I should dispel. There's a lot of people who like to say that, they woke up when they were 8 years old and they decided that they were going to get the Nobel Prize for whatever it is. And it's not true. Each decision point has a myriad of options, and there's a large dose of opportunity, chance, chance posts, chance meetings, and influences in life. And so the only thing I can recommend is having a lot of irons in the fire, or a lot of interests and possibilities, so that when one doesn't mature, you can actually make a course correction and keep going. So with that in mind, I'm going to talk a little bit about regenerative medicine and translational research. So a few diagrams on translational research. The lab bench is an experiment. So let's say this is arcane, impractical, and slow. You can't do experimentation in the patient. It has to be fast, simple, and obviously effective. And there's a huge amount of work which goes into taking it from the lab to the application, regardless of what science, what field you're in. I'm going to talk about medical research and implantation technology. But as Thomas Edison said, it's 1% inspiration and 99% perspiration. I think that he had about 100 people working for him, and he considered that he was the 1% of inspiration, i.e. that all the ideas were his. But it sort of pans out that, the amount of man hours, person hours, and financial investment, you can get to the patent with about 1% of the total requirements to bring the technology out into a commercial application or a clinical application. And timing is, people will start, and timing is very different. It could be a year, it could be 10 years, it could be 100 years. We're working people who've just got approval for nerve growth factor, which was discovered in the late 50s. And so it's been a long time to clinical translation, even though it's a very powerful technology and was the first, nerve growth factor. And so being able to make it is important, and being able to deliver it appropriately, as applies to many, many drugs and many, many regenerative medicine applications. Two paths, I think, fundamentally to translational research. New science is the sort of attractive and the one which is going to win the Nobel Prize. A new discovery which then is applied across a large number of fields, and also specifically, which will change the game. It gives you another piece, you know, another set of pieces of the jigsaw puzzle, and you're suddenly able to complete that jigsaw puzzle where it was impossible to do so before. And you see, you know, various leaps and bounds in product development or clinical development based on a transformational discovery in basic science. The other side is a little more mundane, but is really where the majority of new technology comes from. It's existing technology which is applied to a new field, or existing products even which are applied to different applications. They are literally engineered. There is a problem and the systems are engineered to address that problem using your smorgasbord of technology which you have available to build the systems. But it doesn't matter whether it's electronics or mechanical engineering or bioengineering or molecular biology, that still applies. You are taking elements out of the erector set or Lego set of opportunities and building a new structure. Both pathways produce everything around it. The other thing is that all my colleagues would want me to say on the business side is that it takes finance. And you know, as basic scientists, we're all aware of the need to get grants, etc. But the amount of money which is in a grant, even the substantial grants, is paltry in comparison to what it takes to launch materials or to build a company. We started Integra as a very small company just in wound care and did not make a profit in the mid-90s. And we are now the largest neurosurgical franchise in the world. We managed to buy part of J&J, their neurosurgical business, a few years ago. And so now it is very profitable. And we hope it continues to do so. But you have to pay attention to this side of the business. You have to understand the drivers from the financial folks and the legal folks and the business development folks. You can't just be in splendid isolation as an academic scientist in industry. You really do have to participate and be one of the drivers for building new businesses. So beginning to wrap up on this section, this is a sort of list that you might put down to get a new idea or a new product. All the things you need to do down the left-hand side, and you have all the people who might be involved. It's not a complete list or an exhaustive list, but it will do. The problem is that most people like to think in a linear fashion, and you go from one step to another, and most processes for doing development are structured in that way, and it is not so. This is what it looks like. It's a complete maelstrom of activities which are happening concurrently with feeding into each other, and like snakes and ladders games, you return, you slide, very, very rarely get a ladder. And so things don't work. You have to plan for that. And obviously, system does work because we live in an industrial age with all the benefits of this type of activity around us. But thinking that it's a linear process and that you don't have to understand the entire process from conception to product launch is dangerous. And there are very many traps and pitfalls if you don't work on conceiving of the entire process, because there's stuff which will blindside you, both as an individual and as a team. It's one of those things. It's not simple, and if it was simple, everybody would be doing it and would be successful at it. So a couple of examples. We have Duragen, which is a dural graft, which Kathy mentioned, and Neurogen, nerve guide. So my involvement in these to look at the translational timing is at the time of launch, the Neurogen was 22 years. So we launched it in 2001, and Duragen took 22 months from first concept of bringing it into the clinic and delivering it. So we'll talk about DuraGen first, and it'll give you some background on our collagen technology. So our platform technology at Integra is collagen. It's part of the extracellular matrix, and it's a structural component of the extracellular matrix, as I think many of you will know. And we have a process by which we purify it. So our business in making regenerative matrices is very much like making paper. We take a natural material. In the case of paper, it would be a tree, and we render, which is rendered down to cellulose fibers to make paper, to make white paper. If you're making brown paper bags, it's got a lot of lignin in it as well, because that adds strength and sort of glues the cellulose together. So the analogy for the Integra collagen is that we take a cow tendon and we remove everything except the fibrous type 1 collagen, and we further purify that so that it's compatible cross species, which if you're trying to regenerate. So we take the tendon and we treat it with enzymes and with sodium. It's about the same caustic level as Drano, and we protect it. We salt out the solution first to slightly protect the collagen. But that destroys everything except the type 1 collagen, and it chews the collagen triple helix off from each end as well. So the terminal chains of molecules which are not in the triple helix of collagen, which actually identify it as being a cow or pig or bird, of some sort, it's a species specific. So if you chew that off, you just left with a triple helix, which has the same structure across all chordates. And you then end up with a generic collagen, which will be accepted by any chordate. And we know that. We have studies in fish, in birds, and we have a Komodo dragon in a zoo, which we regenerated its skin, etc., as well as all the mammal models. So that is, if you can't do that, everything else is out of the question, because if you put the material in and instead of encouraging regeneration, you get inflammation, you get scar tissue. And that is not regeneration, it's tissue growth, but it's not. It's an emergency patch the body makes to try and wall things off or to plug a hole. And so we actually slow regeneration by making matrices out of the system. So we take the individual fibers, and you can see the banded structure, which means they're not denatured, and we felt them and cast them into a number of different structures, but primarily a three-dimensional open-pore matrix. And the radius of curvature of these matrices is really important. If you make it too flat, cells don't like it. If you make it too tight, too smaller pore size, they can't get into the matrix. So we have 80 to 90 microns is about the size of the pores, so somewhere around the size of a human hair. And we make these matrices in different constructs. And the amazing thing is that if you implant this matrix, it's recognized as self. Cells grow into it. The material gets revascularized, and they don't, the inflammatory cytokines or things which promote scar tissue are shut down because it's recognized as self. And instead of making scar tissue, the cells enter the matrix and they listen to local cues coming from the surrounding tissues, and they will actually make an appropriate tissue for that location. So it allows to shut off the scarring response and switch on the homeostatic response, because our bodies are being completely renewed all the time. I think the only thing which is not renewed in our lifetime is the DNA in our neurons. Since the neurons are pretty static for most time, there is some regeneration and neurons added, but the majority of them try and last us a lifetime. But their membranes are being turned over all the time. They're rebuilding themselves, but other cells are dying and being replaced throughout the body, so homeostasis. So that's what our tissue regeneration really is recapitulating that process, preventing an emergency response to injury, which is scar tissue, and being allowed tissues to grow. So DuraGen. I had a conversation with the chair of neurosurgery at Philadelphia at the time, Eugene Flam, who's still with us. And we were talking about regenerating spinal cord injuries and doing neural transplantation. And he had read a paper, and basically, can't always fancy regeneration technology to do something really useful like regrow the dura. It's very tiresome to have to suture the dura after a long case. So he wanted to be able to regenerate the dura, to get the dura back, because using materials which were available at the time had a lot of scar tissue encapsulation. But also he'd read a paper from South Africa from James Dillon and his team, where they had developed a onlay graft. So we're entering, the concept is entry here. This is an idea, a requirement. And from somebody who actually knows a lot about it, that's always a good sign. And somebody who's also done their reading and has got some ideas of their own. That's not always necessarily the right idea, but at least they're giving you a direction. Dura grafts, they're used to repair the dura. The dura lies under the cranium. You have to go through the dura to have access to the brain to do neurosurgery. And since the get-go, it's been important, and people realize the importance of suturing the dura. Reports of dural suturing are from the 1700s forward, really, only doing it quite effectively sort of in the 1900s, and particularly after the First World War. American surgeon Harvey Cushing, one of the founders of modern neurosurgery in the US, was treating young men who were perfectly conscious in the First World War, having been injured in the head, but they had a CSF leak. And the majority of them were dead with bacterial meningitis within 10 days. And so he said, if basically thou shalt suture the dura. If you do nothing else in your procedure, you need to close the dura so that the CSF is not leaking out and providing a path for bacteria to get in. So obviously, suturing the dura is very close to the heart of surgeons. That's one of the things they can actually repair, because most of neurosurgery involves sucking bits out or clipping aneurysms, stopping things from, or removing tumors. So actually being able to repair the dura is quite significant, because it's an active act of repair in the patient. But it takes a long time, and at the end of an operation, the patient really needs to be in the neuro ICU to be treated medically. So spending a lot of time harvesting some graft from the patient, some other part of the patient, and then suturing it very carefully in is anything up to an hour or more, depending on the severity of the case or the complexity of the case, is time better spent somewhere else. It's more anesthesia, and the more open time you have in any patient, the more likely you are to get infection, etc. So the team in South Africa had really developed this procedure. It's an onlay graft, and you're done. Cut it to size, you put it on, and you're done. And they'd used a collagen matrix, which was available for other applications, and they had been using it as a temporary way of repairing the dura while they got the patient, the vital signs of the patient up after people with head trauma. And in the 70s and 80s, there were a lot of blunt force head traumas because of the troubles in South Africa, so they got a lot of practice. Collagen is not particularly, has been used in neurosurgery for a long time. A famous neurosurgeon in the US, Peter Giannetta, was working on formaldehyde cross-linked collagen sheets. Kersner Puzo, James Dillon with his collagen matrix as a dural separation layer in 1978. And then Narottam did his PhD thesis in South Africa. So the South African team really took this and ran with it. And everybody else thought it was sort of a weird sort of idea, but let the guys in South Africa get on with it. Well, they did all the clinical work first, and then, well, majority of it first. And then Narottam did some work in primates and demonstrated that you could regenerate. That's what was happening. The dura was being regenerated, and you can see here, this is the matrix, this is the bone of the skull of the cranium, and there is a new dura at the right distance off the cranium being regenerated, and the rest of the matrix gets resolved. We now know if you put 3 or 4 sheets on, you'll still only get 1 dura. You don't get double thickness because it's regeneration. The cells are growing a tissue because of the local cues, and you can put several sheets in to actually prevent adhesion from the injured tissues around the regenerating tissue from sticking to it and causing adhesions. They then went with that animal data to really give them the confidence. They went back and looked at a large number of patients and a 5-year retrospective review in over 450 patients. And they published it. And that was the paper that Eugene Flamm had seen. And Integra made the matrix they were using, but it wasn't intended as an implant. It was intended as a way of protecting surgical instruments. But at the time, that was it. You know, you publish your paper, and if you want to translate the technology, then you have to do something which brings it into the commercial realm. And they did try to do that. They went to various companies, and they were told that it wouldn't work because you couldn't suture it. They said, but it's an onlay graft. Well, nobody knew what an onlay graft was. So every other material, bioengineered material for duroplasty looked like the normal dura. It was tough, flexible, and you could suture it and really crank down on those sutures. And their technology, you not only did not need to suture, but you couldn't, because it was a friable collagen matrix. And so they went to a number of companies, and they were politely shown the door. And I had the meeting with Eugene Flamm. I called Jimmy Van Dellen in South Africa and flew there the next week. And on a handshake, we agreed to move forward. And they gave me all their data without a confidentiality agreement, which is something that you shouldn't do. But I think we were fairly honorable about the whole business, and they're co-inventors of the matrix. And we came back and custom engineered a matrix which could be approved for implantation into the central nervous system based on another material which we had, which already had PMA, the highest level of approval for neurosurgical applications for another reason. And that is why we were able to get it from the flight to South Africa into the clinic in 22 months. And that changed Integra. We became a neurosurgical company as well as a wound care company. And it's half the business currently these days. This is some of the animal data. This is regenerated dura in a pig model. This time point, which is about 90 days out. That's the normal dura there down in the inset, and the regenerated dura is thinner, but it's absolutely histotypic dura. It has all the layers of the dura, the different structures within the dura. So truly tissue regeneration. Not that neurosurgeons care about the regeneration at all. They care that it pairs the dura and you don't get CSF leaks and it's quick. That's what they care about, and rightly so. They don't need to know how it works. It's better if they know how it works, but people don't care about how the fuel injection system, the turbocharger, works on their Porsche in general. They really care that when you put the foot on the gas, it goes like a bat out of hell. So, you know, a lot of the technology in our lives is hidden. You know, hardly anybody knows that you have a fractal antenna in your cell phone, and that's how it's able to connect to the internet, etc. So invisible technology, which I think is something, it's another lesson, is you've made something which is transformational, but the world, in effect, doesn't care about the technology. They care about the thing that it does for them and what it allows them to do. It's a sobering thought for all the work one might have done to bring something to the public. So peripheral nerve, this is much more associated with my life story. I started my PhD with Richard Fisher, and he'd done his fellowship in the US and had said, you know, he really worked on being a really good peripheral nerve surgeon and was still disappointed with the results. So why do nerve repairs do so poorly, considering how profusely the axons regenerate? So axons love to regenerate. They regenerate everywhere. And they regenerate in places which you really don't want them. They make pain in the wrong place. And trying to get them there, like recalcitrant children or herding cats, you getting them into the right place is a real problem. And up to that time, microsurgery was only developed for peripheral nerves in the late 50s, early 60s. by Hanno Milesi in Austria. And so the use of a microscope before then was unheard of, or magnification. People would take a big fat suture and just yank the nerves together and put a stitch in it, like repairing a tendon. Of course, results were highly variable and usually not very good. So use of microsuture, which had been developed for vascular repair, and microscopes which had been developed for vascular repair were applied to peripheral nerves, and with a huge increase in the outcome. However, where you connect the nerves together, they get scar tissue, and the nerves escape out of the suture line and don't go where they're supposed to. And so, how would you best do that? And for those of you not familiar, the neuron is in the spinal cord, or dorsal root ganglion for sensory neurons adjacent to the spinal cord. And the axon extends to the periphery. And when you have a nerve injury, the distal part of the axon is cut, and a bit away from the cord, and the proximal, which is on the brain side or the cord side, regenerates in most cases. So those going to muscle for motor control, the majority of them manage to regenerate to somewhere, but not necessarily to the right muscle. But unfortunately, sensory neurons are much more fragile, and 40% of them die on cutting of their axon. They can't take the loss of the cytoplasm. So you're starting off, from in a race where you've taken up, 100-yard dash where you're now 50 yards behind the start line and wanting to try and catch up. So how to do it better? And we first started looking at, the mechanics of a peripheral nerve and the mechanics of actually suturing it together. So you've got this incredibly delicate system of axons and myelin and Schwann cells, and you're basically trying to mechanically join it together. And it's not optimal. And you can see, the tissues here of all the axon-containing tissues. If you just put one suture in, most of it is exposed to the environment, and blood-brain barrier is not there. And so a lot of scar tissue will form, and you'll get a painful neuroma, which is scar tissue with lots of axons in it, lots of sensory axons in it, and it becomes very tight. So if you just touch it slightly, all those axons fire. So it can be very debilitating. Microsutures are considered, they're terribly fine. They're about a fifth of a human hair in diameter, and quite tricky to use. And there's no feedback in repair. In doing microvascular repair, it either works, clots, or bleeds out of it. And so there's instantaneous feedback on how well you did. Peripheral nerve takes months to years to regenerate back to the target. And so there's no direct feedback. So that makes it very hard for many people to understand, how to improve their technique, because there's no feedback on the technique. So we worked on a lot of ways of trimming the nerves and improving the outcome, and other people have done that. But they're all very complicated, and the complexity is not the moment here to be, as I said earlier, doing an experiment in the operating room. So we came up with the idea of using tubes in the UK, and other people in Sweden, Nuremberg, was using tubes, plastic tubes of silicone. And there were problems with that. They could regenerate, and then nice models for animal studies, but they weren't resorbed. And in people, they would actually cause a huge amount of fibrosis, and even would break the nerve and be extruded out of the wound because of the encapsulation and foreign body response. So, and our own work, we were looking at who players were here, and most people up to that time had been looking at the axons, obviously, because the axons are conducting systems. But we realized that if you didn't have any Schwann cells, which are the helper cells, which make the myelin, which improve the conduction and provide nutrients to the axons, if you didn't have a bridge of Schwann cells, nothing was going to happen. So we concentrated on making something which was simple and something which promoted Schwann cell regeneration, and the axons will come. So the axons of the sheep, the Schwann cells of the sheepdogs, and the path layers. And the axons go where the Schwann cells tell them, quite literally. And so we created in the UK a living tissue by putting a silicone rod in sub-Q in animals and getting tissue response to it, and then taking out the silicone rod, and you can see the stainless steel surgical wire here keeping the lumen open. And if you take that and repair the nerve, the recovery and the ease of repair is actually quite extraordinary. So we worked on that for a while, and that was one of the reasons I came to Boston. Was recruited to work on the collagen nerve guide. And we modified a system which had been developed in the early 80s, which didn't really work, so that it was more like the living nerve tube without the cells in it. And we ended up with a material which, if you implanted, would be repopulate the cells and provide a living environment in the blood-brain barrier for regeneration. And so it's you know, it's partly neuroscience again and partly biomechanics. We worked on how you cross-link this collagen. Normally collagen is very flaccid and falls flat. A vein has been used as a graft, but it collapses because it has no structure. An artery works very nicely, but removing an artery which is 2 to 3 millimeters in diameter from somewhere in the body, it really compromises vascular supply, and you can't do that. So we made a material which had all the characteristics of an artery as far as its ability to rebound after compression and to be resorbed completely over a period of time. We did primate studies, and we showed that I won't go, these are electrophysiology studies. Points to take is the long period of time we followed these, 1,400 days. The dashed line here is the normal. This is the lower 95% confidence interval. And you can see that these repairs were direct suture, graft, and tube. And in this study, the tube performed better than the other two standard methods of repair. And we did these in primates and reported it from the early 90s through to the late 2000s. And that's what we finished the bulk of the animal data in 96, and I came to Integra in 97. But we had to rebuild the manufacturing, and we had to rebuild the cross-linking, and then do the clinical trials in Europe and launch. And so it took us a while. We started in 97, and we launched first in the US in 2001. So 4 years from actually trying to commercialize it. So I don't think it's too bad. But all the work we've done, like I said, in the clinic and in the lab and the animal studies were a tiny amount of the effort which was required to actually launch it. So you don't stop there because it's not perfect. Well, you're not even trying to get to perfect. It's not. It's obviously there's huge room for improvement based on grafting technique. We're using autografts based on what's supposed to be gold standard is direct suture and intubulation, etc. So understanding what's going on in peripheral nerves is a continued challenge, and we have done that through collaborations. And one of these collaborations has produced this material, which is basically all the tissues of a peripheral nerve which have been synthetically manufactured or artificially manufactured. So you have the outer epineurium, which is a support layer, perineurium, which is a blood-brain barrier, and the endoneurium, which is the matrix which supports the Schwann cells and the axons. And it has this counterhelical windings of collagen in the surface, and it has longitudinal collagen fibers and pores in the matrix to simulate the basal lamina of Schwann cells and axons. And so that was the next stage. This was actually invented by us in 1989 and patented that year, a collaboration between MIT and Harvard. The patent long expired because we couldn't make it. You could make 20 of them, and 1 or 2 would turn out right, and you had to select them. If you cannot make it, it's not a product. It has to work every time you press this button and run the machine to make it, or whatever the process it is. So it took a lot of noodling on thermodynamics to make these longitudinal matrices, and we finally cracked it. And it's had FDA approval for about 3 or 4 years, and we're just about to launch it sometime in the next 10 months or so into the clinic. But it was very exciting when we did animal studies with it. This is a normal nerve. This is a blank tube. This is a matrix with just collagen, and this is matrix with our secret sauce, which is a chondroitin 6 sulfate, which is part of the Schwann cell basal lamina. And you can see that the graft and the new matrix, collagen gag matrix, support, look very similar. And this is at a gap where you begin to fail with intubulation. There's just not enough time to fill that gap with Schwann cells, and you don't get very good regeneration. So you get a lot of regeneration, but not profound regeneration, which you want to completely reestablish connections. And it shows that the number of axons which regenerate in an autograft are now the same in the collagen gag. There's no difference between the two. So in this model, it was as good as an autograft for these long gaps. And when you compare the microscopy, number one is the regenerated autograft. The number two is the tube with these islands of matrix. Number three is a collagen matrix. Still islands, but no microvesiculation, no barriers between the two of them. And then the new material, and it's not discernible which of these is which if they're randomly presented to somebody, or even to myself. You usually can pull stuff out, but they're virtually identical, which was really exciting at the time. So working with others, we have three main collaborators in peripheral nerve. Alan Levy at Miami Project to Cure Paralysis at the University of Miami, Raj Mida in Calgary, and Fergal Brian at Royal College of Surgeons of Ireland, and a collaboration with the AMBER program, Advanced Materials and Bioengineering Research Group in Ireland. And just quickly, we've been through the first and second generation, which I've talked to in detail, but the third generation is looking at other materials to put on the matrix to make it more like the basal lamina. We have fibronectin laminin and laminin 2, or mericin. And what we discovered is that in very specific ratios, you could upregulate the expression of nerve growth factor and mimic the environment which was being presented to the Schwann cells, made by the Schwann cells during the process of regeneration. So we're getting up to 6-fold increase in the number of Schwann cells in these matrices being stimulated, and a 2-fold increase in the rate of migration. So very exciting for bridging larger gaps. Though this is some time away from the clinic, but it's the stuff you have to do so that you can get to the clinic eventually. Just published this year, using them back into the matrix. Alan Levy, who's been leading this with the Schwann cell and a combined interest, there is the patient's Schwann cell, well, these are rat Schwann cells infused in the matrix, and they're labeled with S100 so that you can see where all the Schwann cells have gone into the matrix. And so this is now a living graft, and it performs almost identical at large gaps in the animal models. So that's something we can look forward to possibly in about 6 years in the clinic in the US. So that's all from me. I'll leave some time for some questions, and I apologize for the late start. But over to you, Kathy.
Thank you very much. Does anyone have any questions? That was quite impressive, to say the least. Here's a question in the Q&A. It says, to what extent do you think it is helpful, significant, for academic researchers to understand the product development process of biotech and biomedical companies?
If you want to do that, if you want your science to be relevant in the fact that you actually want your science to be translated, it's essential. If you're working in purely basic science where you were following, the molecular biology or some technology or whatever, it's not so pertinent, but it does help, particularly if you discover something which is really useful. I've had friends and colleagues who've taken quite small advances into the clinic and have done both personally and reputationally very well out of it. And I also know people who just had no understanding of how the system worked, who had made really quite major discoveries and actually lost their research grants because of the reaction of industry to what they were doing. So I think understanding how it works is really important. I think it's important in general because it's part of, why do you do science? You can do science just because you're curious, but your curiosity costs a lot of money. It costs both yourself and the society a huge amount to provide the resources by which you do research. And I think you have an obligation that it's relevant.
Excellent guidance. You have another question. It says, what course of study would you recommend for someone interested in the private biotech sector?
So if you want to be management in biotech, then a law degree or a finance degree after your science degree is how you're going to do it. Because the majority of people who are in senior management, either finance, lawyers, or have come up through the marketing side. It's just how industry works. There are very few of us scientists. So there's about 10-to-1 ratio of non-scientists to scientists in our senior management team. So if you want to do science, if you want to do development, then you need to become expert in a field. And it's a bit like practicing Zen. Once you become expert in anything, it transcends to being able to be expert more quickly in anything else. There's a road of self-enlightenment which occurs through a postdoc, a PhD and a postdoc, and academic involvement, which will then allow you to embrace almost any challenge, I think, which industry can throw at you from a technical point of view.
Very thoughtful response. Another question, are the people leading the biological research also often leaders of translational research?
No, it's a very different discipline. So as we said in the previous questions and answers, there are different skills required, and you do have to understand, to be able to do translational research. I mean, true translational research, not just translating some basic science into a potential application in an academic setting, but actually taking the technology and delivering it to a customer at the end, like from, as we say, from cow to patient. That requires you to understand how the system works and what is required. And it understands a basic, understanding of regulatory science, of a regulatory realm, operations, etc., and clinical studies in this field, in bioengineering.
Thank you. Well, there's another question. Are there specific fields of law, finance that are more relevant to biotech?
Not really. Intellectual property law is something which is obviously, in startups and in large corporations. But, how a corporation is structured is basically the same come what may. Whether you're making shoes or cars or biomedical devices, the, chief finance officer is dealing with the same stuff. It's, the opportunity to acquire other companies as well. But it's very broad skill set. And, the science, you get the weird and wonderful scientists, which they keep chained in the back room and feed raw meat, etc., are providing all the technology, but the business is being run by a very standard set of rules. So general, business degrees help a great deal. I think if somebody has a PhD and then they do a business degree, it really helps both them understand what's going on and for the business folks to realize that this is not just a crazy PhD who just wants to do science, that they understand what they're talking about. And that ability to have a foot in both camps allows you to do more translation. It allows you to see into the basic sciences, and it allows you to see sort of upstream into development and be a negotiator between the two to get new things done. And it's very much what I do now is I nurture and chaperone new technology into the company and provide justifications for it. Often when people don't see that they need it, or that it's going to be useful or very profitable for the business. And you have to take it up to a certain point and then start outside of the business and then begin bringing it in. And if you translate too early, you can kill something. If you translate it too late, you miss the opportunity. So it's not simple, but the Dilbert cartoons are funny because they apply to all companies and all groups of people who work together. And that is, I had no experience. I learned my business from hard knocks and coming in and doing stuff from scratch in quite a small company and building it to helping to build it to a large company.
Yeah, wonderful answer. Wonderful answer. Are there any more questions? No. Thank you so much, Dr. Archibald. What a wonderful presentation. The slides were incredible. Absolutely incredible.